that's not right. Let's try that again. Oh, sorry about that. That was a little bit better. Technical difficulties. Okay, so nerves and muscles work really, really closely together with each other, in part because they have what are known as excitable membranes. Okay, so excitable membranes have repolarization and depolarization events. So we basically have changes in voltage across the membrane, right? So it becomes more positive, less positive, that sort of thing. And these changes create something called action potentials. And these are electrical impulses that we think of when nerves are like firing and stuff like that. These are called action potentials. Skeletal muscles, the ones that we've been talking about, contract because they're being stimulated by motor neurons, okay? And so where the neuron meets the muscle, that's known as the neuromuscular junction. And a synapse, we'll talk about those when we get into the nervous system a lot more. A synapse is just where two nerve sections meet, okay? Two different cells meet. So the NMJ, the neuromuscular junction, is the meeting point between the end of a neuron and the beginning of a muscle fiber. And what ends up happening is the axon from the neuron releases a neurotransmitter into something called a synaptic cleft. That's just that space. The neurotransmitter is acetylcholine that binds to special proteins that let sodium in, which changes the voltage across that membrane and starts an action potential. Here is a video clip that illustrates that a little bit more slowly and gets us started with this muscle contraction process.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so once we have sent the electrical signal down the neuron, we have moved into the muscle cell, and we have started that contraction cycle that I just mentioned. Now we get into the actual cycle. So the cycle begins. We have active site exposure, that's the beginning of it. We have the formation of cross bridges. Myosin is going to bind to actin. We're going to have what's known as the power stroke as the myosin head pivots, then the cross bridge is going to detach, reactivating the myosin, and then this process will repeat as often as necessary. And so there's actually an animation that I have. The cross bridge cycle and the actual muscle contraction.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
Okay, so that's basically how muscles work. Now, what's cool about that is that took a long time to talk about it, but like right now I'm talking, I'm using muscles, and you're gesturing with my hands, and I'm using muscles. And so all of that process that we just talked about happens super duper fast. So I don't know. I always think that's pretty cool. You know what, ladies and gentlemen, I think I'll call it there. That's a lot of information to go through. So if you have any questions, send me a remind message, send me an email. Otherwise, I look forward to seeing you all again soon.